Hello. Good morning, everyone, and thank you for being able to join us and taking the time today. We're really excited to have everyone here. This is a really unique event for us at AIP Publishing. You're joining during um, the first inaugural bioscience webinar. And so we are really fortunate to hear lightning style presentations from four distinguished researchers working on the cutting edge delivery of drug delivery, safety, and testing research. So today you'll have the fortune to hear from Sev Sarakaya presenting on behalf of Dr. Horatio Dante Espinoza from Northwestern University in the United States of America. You have Dr. Minjun Sun from Chan Zuckerberg Biohub in Chicago. You'll hear from Dr. Marco Rasponi from the Politecnico di Milano in Italy, and Dr. Ash Agarwal from the University of Miami. So we are very excited. Thank you for joining us, whether it's morning, evening, or afternoon from you, all across the world. This is a true pleasure. Today's conversation is going to be moderated by Dr. Yi Cao of Nanjing University, and he's also the editor-in-chief of Biophysics Reviews. Also on the call is Justin Cooper White. He's the editor-in-chief of APL Bioengineering, and Leslie Yao, he's the editor-in-chief of Biomicrofluidics. These three journals are the cornerstone of AIP Publishing's bio portfolio, and it really is a rare treat for us to be together and celebrate this excellent research across the portfolio. So now I'll turn it over to each of the EICs to introduce themselves and give you a little brief update on the journals, their scopes, and the visions that they've really created and the hopes that they have in servicing you as researchers in the community. So first, I would like our moderator, Dr. Yi Tao, to share a few words about himself.
Good morning, everyone. I'm Yi Cao from Nanjing University and the editor-in-chief of Biophysics Reviews. It's my great pleasure to be the moderator of this webinar. And as we can see from the name of the journal Biophysics Review, it publishes high-quality, cutting-edge reviews in the broad field of biophysics. And not only review papers, we also publish original research papers. And we just checked the recent journal impact factor is 4.5. So I think it's a good review journal and look forward to the submissions. And we are looking forward to the discussions this morning. Thank you.
Thank you, E. Very good hands with E as our moderator. And as he said, Biophysics Review is an excellent journal publishing reviews, research across the biosciences. Next, we have Dr. Justin Cooper White of APL Bioengineering. Justin, if you would like to say a few words.
Sure, thanks, Austin. Good morning, good evening, everyone. Joining in from Australia, it's 11 p.m. over here, so fairly late night for us. Leslie's similarly late. APL Bioengineering has been going for a number of years now. We launched in 2017, and we focus on publishing papers, review articles, perspectives at the intersection of biology, physics, and engineering, and really looking at high impact manuscripts that advance our understanding of physics and engineering of biological systems. We cover a whole range of topical coverage areas that you might expect: biofabrication and bioprinting, biomedical materials, engineered living systems, cell and tissue engineering, regenerative medicine, molecular cell and tissue biomechanics, and systems biology and computational biology. And it's certainly launched into the space of AI and machine learning in each of those areas. And we look forward to seeing your papers whenever you'd like to submit them to APL Bioengineering. And thanks so much for attending the webinar today.
Thank you, Justin. APL Bioengineering has been one of our newer APLX journals, started and it's reached its decade of publications, and it's really been the landing point for the journal and for the portfolio to touch researchers and help you expand the breadth and awareness you have in the bioengineering space. So kudos to Justin. And now I would like to let Dr. Leslie Yao, Biomicrofluidics, say a few words and introduce his journal.
Thanks, Austin. So Leslie Yeo, calling in from Melbourne, Australia. I'm the editor-in-chief of Biomicrofluidics. So BMF exists to, publish the fundamental and applied research in all scope across bio, micro, and nanofluidic and transport phenomena. Now, the contrary to common perception that it has to be bio, micro, and fluidics, it's actually or. So we publish across all that, all throughout the ranges. So whether it's biofluidics or microfluidics or nanofluidics, nanotransport, we accept all that papers. And what we like to do is being a society-based journal, we like to partner with, the community to publish strong papers that are not just show and tell, in, devices, but also strong in fundamental science. And that's what we've been strong and known for. So thank you for joining us. We really appreciate it and really excited about this first webinar. Thanks, everyone.
Thank you so much, Leslie. And as you see, we have the opportunity to poll just popped up on your page for most of you as participants. And we just wanted to get a sense of what brought you to the webinar. We're excited to have you here, no matter what time zone you're joining from, and would love to know which of the journals you're most familiar with, or if you're not familiar with any of them at all. Just really appreciate your time, and we'll want to do everything we can to make sure you get the information about the journals that matter most to you. So with that, as you populate those answers, feel free to take your time. They'll be live and updated throughout the course of this webinar. So I won't delay any further. I know you're all very eager to hear from our speakers. So with that said, I will allow you to introduce our first speakers for the day.
The first speaker will be Dr. Sang. Sorry. Will be second. So I need to the first talk will start from Dr. Minjin Sang from Chan Zuckerberg Biohub Chicago. He leads the microtissue signal dynamics group. His research integrates microfluidics, single-cell analysis, and systems biology to understand how immune cells process dynamic signal in space and time. Today, he will discuss advanced microfluidic technology for precise intracellular delivery, genome editing, and the real-time analysis of living cells. The floor is yours, please.
Okay. Can you see my screen?
Yes, we can.
Yes. All right. Cool. Yeah. Thank you for the introductions and thank you for this opportunity. So my lab is in Biohub Chicago, and my lab is especially specialized in developing the microfluidic devices for the tissues and controls very tightly controls the fluids inside. And on the other hand, Dr. Espinosa from Northwestern, his lab is specialized in the cytokine measurements through the plasmonic sensing. So we are collaborating together to enable something very cool. That is so our ultimate goal, by combining our technologies, we are trying to build an integrated platform where from my lab, I can leverage high throughput microfluidics and to have like a tissues or organoids growing in many tiny chambers, and then to control the fluid and extract a little bit of each fluid from each samples, and then send them to another chip where the tools or the devices from the Dr. Espinosa can use his plasmonic sensing technologies to measure the cytokines. So through the microfluidic chips, I can control the inputs. I can apply many like a TNF alpha or LPS or any combinatories of the drugs. So these are the inputs, and then these can come in different combinations or different dynamics. And then I can address each chamber with these different inputs. And then the outputs, I can do the like real-time 3D imaging. At the same time, send a little bit of fluid from each sample to the another chip that has a plasmonic sensing. So if this is possible, we can monitor like hundreds of tissues. We can monitor how they respond to this stimulus dynamics or the combinatory stimuli, and we can measure like the fluorescence changes or cell migration happening in the tissues. At the same time, we can measure the cytokine secretion dynamics in the real time as well. So yeah, this is our dream. And so this is divided into two phases. The first phase is high throughput microfluidics. And because it's a high throughput, and then there are many like automations necessary to run these experiments accurately. So my lab has developed this microfluidic platform where it has a lot of arrays of chambers and also a lot of controls to like mix the input media and then send them to specific chambers or to change the medium condition or the cytokine conditions in each chambers like continuously. So the left picture shows the high throughput platform that enables this. And on the top of that, because we also want to get more data through like a little bit of so we want to print the tissues in a way that we want. So if we just see the cells, the amount of cells sitting in each chamber will be different. How they form the organoid, what the shape can be different. But that data will be difficult to interpret. So we have our own custom like robotic pipette system, so we can actually print or see the organoid in a specific uniform shape that we want. And also, I mean, the last piece that's necessary is the 3D imaging. So we have a kind of custom confocal. So I'm about to purchase a spinning disk confocal to facilitate the high throughput imaging as well. But for now, we have we have a confocal system that will enable the 3D imaging from each samples in each chamber in the chip. And just to briefly show you how the system works. The left side shows so all these blue little part is like a small chamber that we have organoid or tissues in it, and all these the holes on the left side is where the input media will be plugged in. It can be a TNF, it can be LPS, it can be IL-12 or some drugs that you can just imagine. And there are these many fine red lines which has variables to control which of these inputs will be mixed and then send it to each chambers. So these are all controlled by the computers, and then these all can be automated. And then the one in the middle shows how the tissue printing works. And then the image on the right side shows that how it looks like when it's printed. So on the top, you see that there's a chamber um, filled with so this black part is a deep chamber. It has a thickness of about 500 micrometer or so. And then the pictures on the bottom images on the bottom shows that how it looks like when the organoid is seated. And this Seb will take on.
Yeah, thanks, Minjun. So yeah, here my name is Serket Sarikaya. I'm a postdoc in Professor Espinosa's lab. Unfortunately, he had a time conflict, so he's not able to join. So this is a collaborative project with Minjun's group and our group at Northwestern. So here we have a microfluidic device with gold plasmonic sensor. So we functionalize this sensor for like proteins of interest. And when there is a binding to the sensor surface, the sensor reacts it optically. So we track the optical reaction in situ with microscopy and camera. So this microfluidic device allows the cells to stay alive for days while we do like continuously read the sensor outputs. So we can track like individual inflammatory responses with high spatial and temporal resolution. At the same time, like we can track the morphological changes and we can correlate these morphological changes with the secretions. You can go to next slide, Minju. Yeah, so before any, live cell work, so we calibrated the sensor against like known concentration of recombinant like cytokines. So these plots show the real-time like binding responses for TNF alpha and IL-6 from 100 to 500 picomolar. The signal is like rises in a clean and concentration dependent way. So when we when we plot response against like concentration, both cytokines like give a linear fit with a high R square. So that means like we can convert this optical signal directly into absolute like protein concentration. It's an important to back calculate the cell responses into the amount of secretion they have. Next one. Yeah, so with that calibration, we moved to live mouse astrocytes simulated with LPS, which is a molecule that can stimulate inflammatory responses. So here is like showing a bright field images of the cells in device and corresponding final intensity increase map at end of the experiment. So we first identify the location of the cells and track cell position by recording and processing intensity changes within that specific cell region. So also like we show the intensity increase over time. And in this graph, so the thicker line is the median values of the whole cells of the whole region. So we see a scatter in the response that we believe it is a biological cell-to-cell variability. So also we validated this platform against ELISA by normalizing the responses with the number of cells. So the device and ELISA response is overlaid on the graph, so they track each other over 24 hours. So the slope is like they agree each other. So we want to show the ELISA, which is a gold standard method, and our method agrees. Yeah, next one. Yeah, this slide basically brings the full platform together. On the microfluidic side, so the microfluidic device handles the cells and reagent purging and drug and ligand delivery. And also the array of tissue cultures for like parallel and high throughput cell culturing and acquisition, data acquisition. So the robotic pipette brings the tissues and organoids directly on top of the chambers, and the entire stage sits on a microscope in a controlled atmosphere. So the other optical side, we have this dual measurement system within the same field of view. We have two outputs from spectrometer and also the camera. So we can get sensor data, and also at the same time we can have the morphological track the location of the cells and obtain the morphological changes. So actually, this is a unique combination of everything. That combination is exactly what led us to measure whole like tissue responses to dynamic and combined like stimuli at high throughput, which is our original goal.
Okay.
Yeah, this is a very much like a team effort from BioHub Chicago and BioHub San Francisco and our group from Northwestern. Thank you, everyone that's shown here for experimental and engineering work, and for Chan Zuckerberg BioHub for the funding.
Thank you, Dr. San and Dr. Sarikaya, for the excellent presentation. I especially appreciate the integration of delivery technologies with real-time cellular monitoring. This type of platform could be very powerful for understanding complex biological responses. And again, I invite the audience to continue submitting questions to the chat box. We will move to the next speaker. Next speaker will be Professor Michael Rasponi. Professor Rasponi is a full professor of bioengineering at Politecnico of Milano, where he heads the MIMIC lab within the Department of Electronics, Information, and Bioengineering. His research focuses on organ-on-chip platforms that integrate microfluidics, biomechanics, and electronic stimulation and human cell biology to model cardiac, musculoskeletal, gastrointestinal, and pulmonary tissues with a strong translational orientation towards drug discovery and safety assessment. He is co-founder of BioMixXer, a polytechnical dynamical spin-off translating organ-on-chip technology into pharma R&D workflows, and currently serves as the chair of the European Organ-on-Chip Society. So we're delighted to have you with us. Please go ahead, Dr. Michael Rasponi.
Okay, let me share the screen. Can you see it?
No, not yet.
Okay, now I think so.
Yeah, please.
Okay, yeah, thank you. Thank you for the introduction, and thank you everybody for attending this webinar. So in the next only two slides, I will try to guide you through research that we published on APL bioengineering in 2018, so 8 years ago. And the rationale behind this publication was a biological question, so how to improve the maturation and the functionality of a miniaturized cardiac model. And at that point, that year, actually 2 years before, we had a publication already submitted. I will try to share that with a video in which we developed a platform made of PDMS, able to, here you see 3 chambers, all actuated with the same line. And each of them was filled with a gel containing cells, cardiac cells, in a 3D environment, in a fibrin gel. And separated by 2 side channels here in blue, serving as vessels. And once polymerized, the actuation chamber was actually used to provide a mechanical cyclic loading, which actually turned into uniaxial strain to cells. And that turns like mechanical training to cells. The cell source at that time was neonatal red cardiomyocytes in a ratio of 70 to 30% cardiomyocytes and stromal cells, mostly fibroblasts. So the idea, with that idea, we went on adding, we kept all the essential elements which proved to be useful, and we added another element, which were 2 electrical electrodes, stainless steel electrodes, which were fitting inside the flanking channels, so surrounding the gel itself. And the first step, and the aim was to deliver not only a mechanical stimulation to cells, but also an electrical stimulation and possibly a combined electromechanical stimulation. The first question was about the electric field that was needed. So we did a finite element simulation, a model, and then we used 2 different types of stimulation. One basically exploring what was the state of the art at the time. So the traditional one was to provide a 5 volts per centimeter electric field, and the other one was to control the current, providing a higher current density, which was 74 milliamp on the square centimeter. So the results, the preliminary results, you can see them here on the right. So the control providing a calcium dye demonstrated that after 5 days of culture, so no mechanical, neither electrical stimulation, the cells were kind of sporadic beating, while with pure electric field, there was a synchronization of the cells. And the best result was achieved with 5 volts per centimeter of electric field, so a mild field, which was following a pacing of 1 hertz. So with that, we went on in our study, and we tried to see whether the electrical stimulation was better or not of the control, of course, and the mechanical stimulation, or if coupling them was actually beneficial for the cardiac tissue. So what we got was an increase in all the stimulations, so electrical, mechanical, or coupled, in terms of excitation threshold, so the voltage that you need to apply to for your cardiac tissue to follow your pacing. And also we had an increase, a slight increase of the maximum capture rate. So the maximum frequency that your cardiac tissue will follow your stimulation, external stimulation. Also the protein, gene protein level, we had actually some not univocal results. I have to admit that the statistics at that time was not that great. But we could actually see and add a confirmation on what we saw functional in the functional way. So we had increase of troponin T and connexin 43, which is a sign of a better electrical connection of cells in both electrical and mechanical stimulation. And we had an increase of the maturation in terms of ratio of cardiac myosin 7 over myosin 6. So that was basically our unhinged point for our research, and I'd like to show you the roadmap of our research in the field, which, as I said in the beginning, was starting in 2016. Then we had this paper published adding electromechanical stimulation on our cardiac tissues. And then later on, we moved from a rat cardiomyocyte source to a human iPS-derived cardiomyocyte model. And that actually was way better in terms of maturation and functionality. Even because the gene levels were much more clear on humans as compared to rodent models. And after that, we went on using and exploiting the model to test drugs and molecules in cardiac safety environment. That was also a topic of the spin-off, as was introduced, the spin-off of Polytechnic Biomimics. And recently, our current research is on 2 sides. One is to use the model, to explore the model for cardiac safety purposes, from QT prolongation to arrhythmic detection. And the other one is to complexify a bit the model from the biological point of view, as we are adding endothelial cells, which are actually enhancing furthermore the maturation and the synchronization of cells, and the macrophages as well, which are as well contributing in vivo, and we are observing that in vivo as well, in vitro as well, on the stabilization of the in the long term of the cardiac model. Yeah, with that, I'm done with the presentation, and I of course invite you to ask questions either on this or on other topics.
Thank you, Marco. I think you're on mute.
Sorry. Thank you, Rasponi, for the very exciting talk. Yeah, so really very fascinating, from the heart-on-chip tool to real practical preclinical drug screening, and very useful. And I think our audience will put their questions on the chat, and we will read their questions later. So we will move to the next speaker. Next speaker will be Professor Ash Agaro, a professor of biomedical engineering and a director of engineering and applied physics for the Desai SETI Urology Institute at the University of Miami. He co-directs Engineering Career Cures, a collaborative initiative of the Sylvester Comprehensive Cancer Center and the College of Engineering. His undergrad degree from Indian Institute of Technology and his PhD from University of Florida are both in material science and engineering. He then gathered postdoc research experience in biomedical engineering at Columbia University and at the Wyss Institute for Biologically Inspired Engineering at Harvard University. His expertise lies in the engineering of physiologically relevant fluidic platforms that serve organoids on chip, live organ slice, and recently organ perfusion, preservation, and transportation for whole human eyes. So please join me to welcome Professor Agarwal. Please begin your presentation.
Okay, thank you, Yi. Thanks for the invitation, AIP. It's really an honor to present and also be in the same room with, the stewards of science like Professor White and Professor Yeo, who've been, it's really nice when journals are led by career scientists, and because they understand how science is done and can manage the reviews much better. So I really appreciate journals which are headed by, active researchers. And I've had an awesome experience. I guess I've served as a reviewer for both of them. And then also Dr. Rasponi set up the field really well. I don't have any data slides because this was supposed to be just a flavor of what we do. So I have a very high level overview of what we do. The field used to be called tissue chips and then organs on chips, and now it's clubbed into what in the US at least it's called new approach methodology. And I'll just give you an overview, assuming that you have enough of a background on what organs on chips are, on what my lab does and how we think it distinguishes itself from the rest of the field. So we are specifically focused on very large volume microfluidics. In fact, you can't even call them microfluidics. These are plastic-based, non-PDMS pieces that are not bonded together the way PDMS is permanently bonded to glass or other PDMS. And these plastic pieces form the bottom of a chip and a top of a chip, and then fit into each other like interlocking pieces of Lego. When they are clamped together using mechanical forces, I'm not showing the clamp here, they also sandwich between them these compressible rubber gaskets shown in green that snake around each of those channels. And those compressible gaskets under mechanical compression would create the fluidic seal. And now you have independent channels in which you can manually place the biologies in these belts that we micro machine into plastic, and then a fluidic inlet and a fluidic outlet. The inlet and the outlet ports connect to the cassette in a way that the clamping pressure of the cassette brings into contact the inlet and the outlet of the chip. So you solve in this design multiple problems that PDMS suffers from, but the most basic one is PDMS does not allow for you to create very large clearance devices and still ensure laminar flow. So what the only secret sauce behind these chips is that despite having clearances that with Reynolds numbers calculations would not ensure laminar flow, we still sort of ensure a laminar-like flow using geometric features that we machine into the device so that the fluid path remains linear. And that allows for you to place up to 2000 microns or 2 millimeter big organoids in them. So what we want to solve for is the perfusion and the imaging problem of organoids, which usually are done in static systems, or worse, if they're embedded in hydrogels, if they're embedded in hydrogels, then they even need more perfusion and transport. So these organoids can be manually placed in them. And we've, over the last 10 years at the University of Miami, done a lot of work in mostly islet immune interactions for type 1 diabetes, where we place pancreatic islets and we can flow in immune cells into these channels and watch an islet immune attack occur in real time. Again, this is all PDMS free, so it solves for drug absorption problems that PDMS is notorious for. And me and obviously many other labs have developed now a full menu of different organ chips, and the hope and the dream is to do multi-organ connection and get a systemic level response. We just today got a paper accepted in Advanced Science where we've created an immunized organ chip. So immune competent organs and chips is also where the field is heading towards. So over the last 10, 15 years, since the work really got attention by having a kickstart through major funding at MIT and Harvard, and I was lucky enough to be at Harvard at that point working on heart on a chip. We have now progressed towards creating more and more complex chips to doing the hard part of how do we translate this into real impact. And the way that's happening is, excuse me, let me move to the next slide, is really a lot of external factors that are driving the excitement around this field. And the external factors have to do with regulatory law. So in the US anyway, and Europe is ahead of US in that respect, the FDA Modernization Act 2.0 that was passed a couple of years ago now does not require for sponsors who want a clinical trial approval to submit any animal data in lieu of getting approval for human clinical phase 1 testing. So, a lot of excitement happened around, you can read this as you don't need animals anymore, but the hard part is you still have to prove that the assay that you want to submit the data from in lieu of an animal has to stand up to the validation for the same context of use in which you would have used an animal. So if you would have used, let's say, 100 rats or 100 mice for proving that your drug is not liver toxic, for that very specific liver toxicity assay, your liver chip needs to stand up to, needs to be qualified for the same readout and in the same context. And so even though there's a lot of excitement, we wrote this very sober piece in Nature Communications a couple of years ago now, where we said, let's not have organs and chips go through the same typical hype cycle, where whenever a new technology comes, there's a big hype, there's early adopters. But then this happened with microfluidics in early 90s. There is a trough of disillusion where you realize that this is not going to solve every problem. And it's only through very slow and sustained efforts that you find a new plateau. This hype cycle for new technologies is taught to business school students. And so we took the same approach and applied it towards organs and chips and really laid down the pathway to avoid that trough of disillusionment. And that pathway has to do with a lot of interactions, such as like in this webinar, where you have audiences from many different specialties talk to each other. So an engineer will keep designing more and more complex systems, but that system has to be usable by a biologist who often don't like complexity. And then more importantly, it has to be in communication with industry as well as regulatory bodies on what it would take to replace the drug testing paradigm that the industry uses or the assays that the regulatory bodies such as the FDA accept the data from. So with that, I'll invite you to look up the papers we've published, but I'll stop here as sort of a background on the field. Thank you very much.
Thank you, Ashu. Thank you all speakers, outstanding presentations. Actually, it's such a coincidence. Our presentations are focused on organ on the chip or tissue on the chip. So basically, that means this technology is really very important for drug delivery or for drug screening. So I think for the discussion part, I will ask all of you, what will be the most challenging part for this technology go from individual laboratory to clinical use in the future, and how far can we see the real applications of this technology in drug delivery field. And I'd like to ask Dr. Sang to give your opinion first.
Yeah. Yeah. So for since I'm working with the high throughput chips, which has a lot of chambers and then simultaneously monitor many response, I think yeah, this technology can be used to answer many like biological questions. Like I mean, especially biology. Even though let's say you have a skin tissue disease like psoriasis or melanoma, each patient the cause is different, cure is different. Even though you use the same like drugs to cure them, sometimes it works, sometimes it does not work. And then so the high throughput or microfluidic device that we are all developing. I mean, like this platform we are developing is going to be important in answering how the stochasticity, how the like different biology, different biological contexts can be tested and then can be measured and then find out the core mechanism. And all these data can be used to find like a better like a therapeutics at the tailored at the personal like a level.
Okay, thank you. So then Dr. Sarikaya.
Yeah. So I think biggest challenge from lab to like clinical transition is not simply making the device like sensitive or more innovative. It's like it is showing that it's robust, reproducible, clinically useful in real patients. But in the lab, so conditions are usually extremely controlled and like clean buffers, no concentrations, optimized protocols. But like in the real world environment, that's not the case. So the device like may behave like slightly different from like batch to batch. So also like different users like may handle like the essays like differently. So I think yeah, that's the one of the biggest like core challenge. Can the technology, give accurate and consistent results across many patients, many devices for a longer days? Yeah, I think the biggest challenge is like showing the technique is robust, reproducible, and useful, and tried in many patients and samples.
Yeah, thank you. So then we move to Professor Rasponi.
Yeah, thank you for the question. I think I agree with both my colleagues in for different reasons. So I would say that the adoption can be divided in adoption for collaborators and partners, so out of the lab, but just next door. So, for example, in hospitals or in other uses where the clinical question is there. So, in that case, I think the uh, easiness of use, so the robustness, reproducibility of the device is the key point because it's the first time the device goes in hands which are not made engineers hands. So biologists need to use it as much as possible in an easy way. For the adoption in the larger extent, so the adoption of the models, the biological models, then um, uh, that may occur only when there is a qualification of the model itself. So that brings the topic of the context of use, which is very common in the NIM field. So every device can be qualified every time for a specific context of use. So for example, for the heart, despite the cardiac model is a cardiac model, it can do QT prolongation. It can do arrhythmic event, fibrosis or whatever. If I qualify it, so after validation, which is scientific, but also qualification, which is pharma mediated for a specific context of use, so the QT prolongation, then that model would be only usable and used in that context of use. If I want to change the context of use, then I have to go back to the basic ingredients of the biology and start over the qualification. So I think there are different things to be taught, and most of them need to be taught in the beginning of the design process.
You. So we then move to Professor Aguar.
Yeah, I think everyone's saying the same things now, so I'm happy to repeat it. A sort of more or less nice way of saying what Dr. Rasponi was saying is the motto we have in the lab is make the chips simple enough that even a biologist can use it. It's not out of disrespect to biologists, but essentially they're just not they're not used to having any complexity. They want to follow protocol, and it's often not the chip design itself; it's the operation of the chip that's more complex. So the chip can be simple enough, but if you need a full suite of fluid handlers and valves and pumps and manifolds to operate the chip, that is often underlooked, and I would argue underfunded, at least in the US as well. Like developing a fluidic control architecture, if you write that into a grant, there is no study section that will be interested in funding it, because it's just not cool enough, because it's not new science. It's a tool development. So not enough attention on operating the tool, operating the chip is either from funding or from development. And to the second point, that also queuing off of what Marco was saying, just the example of cardiotox, it's unbelievable, right? Like every drug shows cardiotoxicity, and the only assay is that you transfect the CHO cells with the ion channel that has the that you can show the QT prolongation on. That's on a single ion channel level readout, and the next readout, at least in the FDA regulatory law, is dogs. You go right from a single ion channel to a complete large animal. And there isn't any tissue level readout that's fully validated today by the FDA when arrhythmia is an emergent property of multiple cells. So if the risk, is if the point is to show a pro-arrhythmogenic propensity of a drug, I would argue there is a huge gap in the dimensions of where you can go from a single ion channel to a full organism, a full animal. And that gap is being filled through efforts by Dr. Rasponi and others who are developing those systems. And that's such a neat, clear context of use that these devices can fill. So I'm very bullish around, especially heart on a chip and liver on a chip for showing toxicity, for revealing human toxicity.
Thank you, Ashu. So thank you all for the answers. So we have a question from the audience. I will read it. So I think it's a audience from Iraq. So he wrote, in his field of coordination chemistry, drug delivery system plays a crucial role in advanced drug delivery systems through the synthesis of therapeutic metal complex such as platinum, silver, and the copper complex, controlling the stability of metal complexes and regulating their drug release behavior, thereby improving therapeutic efficacy. Conjugated metal complex with nanoparticles or polymer carriers to enhance target drug delivery to cancer cells, increase bioavailability, and minimize system toxicity. I think it's just a comment. So basically, I think with the advanced organ on the chip technology, those systems can also be tested with those organ on chip platforms for sure. So I think all your talk will inspire the audience to use this. So I will move to another question I have. So basically now you see the artificial intelligence and the machine learning have been widely used in almost every field. So how do you see this technology can be used in drug delivery? How will this technology maybe bring something new or even revolutionize drug delivery, especially we have very diverse samples or the results from different technologies, whether they can be combined or can be the data can be synthesized using AI technology. I will start with Professor Song, please.
Yes. So I'm not really chemistry, and drug delivery is a little bit out of my focus. But I do know that there are many microfluidic devices that's used for testing drug deliveries, like droplet generator for mixing many different components or like small matters, and then testing how it goes inside a cell, or I mean something like that. I mean at the end. So with microfluidic device, I mean because it's controlling very small fluid and then very precisely, I think it can be used to create all these kind of different combinations you can think about and then monitor them. And to answer your questions, Dr. Kao, the AI is is already like changing many things in the field, and then especially in the biology. I mean, especially in the biohub, I would say that one of the goal is to make a virtual like immune system or virtual cell. So this is basically rather than solving, I mean, establishing the equations, the differential equations, and then establishing the signaling networks that's happening inside a cell or gene regulatory networks. But now the approach seems like it has a lot. We have a lot of data, and the technology has been developed so much that it can generate a lot of data also simultaneously in one or two like trials as well. So once we have these 10,000 data from these samples, then we have multiple like inputs through the high throughput devices that are making or that while other people are making. And we can use the AI model to connect this input to output. It's kind of a black box right now, but AI can kind of link between these two, especially, the biology is very complex and then it's a very stochastic as well. And so it can be used to kind of help to guess what's wrong with these patients, and then what's the best like combinations of the drugs or the drug delivery systems that can be used.
Thank you. So now we move to Dr. Sarikaya.
Yeah, I agree with Dr. San. So the AI like enables a lot of things that we cannot even think of before. So, but like AI or machine learning will not replace the real experimentation, like in terms of like drug delivery, also the other type of research too. But the AI can make them like much more efficient, like by guiding the formulation design, like doing the experimental design, even like predicting some of the responses. We can like remove them from the experimentation from the get-go. So that enables like more like personalized, like feedback control, like delivery systems. But the key challenge, I believe, is like still like building like high quality experimental data set that makes those models like more trustworthy and clinically useful.
Thank you. So I'd like to hear from Professor
Oh,
Rasponi. Thank you.
yeah, of course, I agree on the use of the AI. But another way it's impacting, at least with me, is I'm more and more challenging my ideas, designing experiments on readouts which make sense, on literature which is published. So it's like conversating with a very smart collaborator, which has a lot of information from the literature. And as a first attempt, when you plan a new experiment to target a possible biological question, it's becoming very useful because it doesn't, it's not, the AI tools are not that wrong anymore on AI, on literature search. So that is something that is actually changing the way we as a group plan our experiments. And the second is again related to cardiac. I'm becoming annoying, I know, but there are tools which try to use.
Sorry.
I believe we might have lost Marco's audio.
This feedback loop is not easy because the biological transfer function is complex, but with AI, there are some ways to simplify that. And so to get what? At the end, a model which is always uh, similar to the target one. So having an endpoint, which is a starting point for drug administration, which is always kind of standardized.
Yeah, then so Professor Agua.
Yeah, so I think everyone's saying the same thing almost now. But the way I think about AI also is it's breaking barriers between silos, right? It's really like if the questions that if the harder answers are if finding hard answers is important, right, to create the next blockbuster drug, it probably won't come from one area of expertise, right? So because the body is so complex and the complex diseases, the most devastating diseases are the ones that affect multiple organ systems that interact with each other in pathogenesis. That becomes really challenging to study when the science, the way it is set up, at least in the US, you can think about NIH has 30 or 35 different institutes, and you can pick your tissue of choice or disease of choice, and there's an institute for that. But there is not an institute that forces people to not just look into what that particular field looks into, but actually learn from information that might be trapped in a different silo, and it could take decades to surface. And what AI now allows people to do is to rapidly access the information that's buried in silos they would have never visited. And that connection, that key scientific connection, is left up to chance, right? Like you meet in a happy hour, or you meet at a water cooler, and that's when those interactions happen. I just think those interactions are enabled by AI in a way that I hadn't appreciated before. And then in the same timeline, the organ chips are becoming more and more complex, and you're adding more and more organs that can talk to each other. And I just think it's the combination of both accessing information in silos very far away from your area of expertise, but also the chips becoming more and more complex, that you can now use these very powerful systems to generate hypotheses that need information from multiple silos and tools which are also from multiple labs together to answer those questions. And that would be very interesting.
Thank you. So let's ask the last question. So, do you have any comments for early career researchers? I think anyone can answer it
I
if
think
you.
just it's a very uncomfortable space for a new research. It's both very exciting but very uncomfortable as well. Like already when we had to train people, and especially in organs and chips, some of them would just be like a deer in headlights, right? Like they just think, oh, I have to learn biology, I have to learn the physics of microfluidics, I have to learn fabrication, I have to learn CAD modeling, I have to learn, computational fluid dynamics. And now you add on the AI, and there's just no excuse to be afraid of complexity. But I just think this is also a very rich training environment for early careers. So the only advice is don't be afraid of complexity and dive in.
Yeah, maybe I can follow up on this because yeah, I think one thing that if a young researcher wants to really work in the field, I would suggest to become like bilingual in engineering and biology, because the this field, at least the NAM field, is very exciting. And one of the reasons that makes the field very exciting is that there is room on the table to speak at the same level with the doctors, biologists, engineers, and everybody in the field. So there are no hierarchical structures at the moment. So it's important to understand exactly what are the issues and what are the possible solutions. And as a follow-up on the complexity, never get in love with the chip. The chip is just a mean to achieve a goal. It's not the final destination of the research.
Yeah, thank you. So we are approaching the end of the hour, so we have to wrap up. So on behalf of AIP Publishing and the participating journals, API Bioengineering, Biomicrofluidics, and the news. Oh, so you have, sorry, so we are unable to do more discussion. Sorry for that. So I would like to sincerely thank all our speakers for sharing their exciting work and the thoughtful perspectives. I also thank our audience for joining us, for contributing excellent questions. Today's discussion has clearly shown that drug delivery is no longer a single discipline problem. It requires the integration of engineering, materials, biology, biophysics, microfluidics, sensing, and the translational medicine. I hope this webinar has provided useful insights into both the current state and the future direction of this field. Thank you again for attending the inaugural Bioscience Research Spotlight webinar. We look forward to seeing you at future sessions. And finally, we'd like to thank Austin for the coordination. Thank you all. Have a wonderful day.
Thank you, everyone.
Thank you.
Thank you.
Thank you.
Bye-bye.
Thank you. Bye.
Justin.